there have been some studies looking at combinations of PI3 kinase inhibitors and other treatment, CDK4/6 inhibitors, et cetera. It seems as though most of those have met with too much toxicity, so we'll see where that goes. But there's interest in using PI3 kinase inhibitors in other settings too. Priyanka, I know that you've led some of the work in that area.
Yes, so we evaluated the alpha-specific PI3 kinase inhibitor with nab-paclitaxel in metastatic HER2-negative breast cancer, and saw some encouraging efficacy in a phase 2 trial. And like you, Hope, said that we recommended prophylaxis with antihistaminics for most of our patients, and that led to really low rate and manageable rash situation. We also kind of asked our patients to have diabetic education right up front, at least in the context of that trial. And I think that helped quite a bit. If we can teach patients ahead of time how to control carbohydrate, that's helpful. And, you know, we also have some preclinical data that suggests that some sort of ketogenic diet, low carbohydrate intake might add to the efficacy of kinase inhibitors. Of course, we don't know that from clinical trials, but certainly in terms of being able to tolerate the drug, that helps.
Yeah, I know there is some concern that giving insulin might abrogate to some degree the effects, and certainly that's of PI3 kinase inhibitors, although seen in animal models, we really don't have any idea about that in the human setting, I would say. And, you know, you have to treat the diabetes. So, but there is interest in the oral agents like the SGLT2 inhibitors as well. So that'll be interesting to see. We want to get John involved in his comments in this area because it's so important for all of us, and also some feedback from all of you, and we hear what he has to say. From a formulary or medical policy perspective, John, how are you differentiating breast cancer at various stages of disease progression? So, for example, how do you decide on CDK4/6 inhibitors first in everybody, PIK3CA mutations? What evidence is needed to support the use outside of the label? And when you have multiple agents in the same class, like CDK4/6 inhibitors, how do you decide which one is okay to use?
Well, there are a lot of questions in there. Let me first step back and say that I think most payers have agreed that they're going to follow NCCN guidelines in choosing which therapies to cover. There are some payers in some states that don't follow NCCN guidelines, but most payers have agreed to use NCCN. Many states, almost 30 states, have mandates to cover FDA-approved cancer therapies. Certainly Medicare and Medicare Advantage plans have to follow the NCCN guidelines or another compendia. Their drugs are in a protected class. Cancer drugs are in a protected class. So I think the question more commonly is not whether or not to cover therapies, but under what circumstances. I think the advent of molecular diagnostics has really helped us increase the probability that patients get on the therapy that's most appropriate for the cancer type. One of the common questions is, can I get paid for doing molecular diagnostic testing? And interestingly, in patients who need IHC testing, for example, nobody's evaluating those. And most commonly, payers aren't looking at single gene mutations. So if you're looking for a single gene mutation like a PIK3CA mutation, those are covered. It's typically when you get into these very costly next generation sequencing tests. So for example, if you have to do a liquid biopsy because you don't have tissue and have to use a Guardant or a Foundation Medicine test, that those tests are prior authorized to determine that they're medically appropriate. Now, interestingly, I work in a regional delivery system. We don't perform these tests locally. I think everyone here probably does tests in their own institution and may or may not be concerned about getting testing covered. But I think that's a significant issue, and one that we have been on the forefront of trying to ensure that people get tested so that we can make sure that they're on the therapy that's most appropriate given their tumor mutation status and stage of disease. So to come back to your questions, I think for patients who are PR positive, if a provider chooses to use a given CDK4/6, I think I said that wrong, a CDK4/6 inhibitor, that's their prerogative to use. And it's also their prerogative in most delivery systems for which one to use. There was an indirect analysis performed last year, published in Breast Cancer Research and Treatment, that compared all 3 CD4K6 inhibitors and showed that they were equal efficacy in first and second line. So a priori, we as a plan don't have a preference over which one was used. There are some differences in the toxicities. I think abemaciclib had higher diabetes or higher diarrhea rates, and Kisqali has higher rates of QTc prolongation. So in those circumstances, it would be hard to tell a provider that you can't use palbociclib first line when the others have toxicities. There are many health plans today that have incentive programs or prior authorization programs around these drugs to try and steer providers to a select drug, but there aren't many circumstances that I'm aware of where a provider is precluded from using their preferred CDK4/6 inhibitor. With that said, in the traditional Medicare population, which many of these patients are traditional Medicare beneficiaries, provider is free to choose whichever regimen they prefer.
That's actually really great to hear. Of course, we do know that there isn't head-to-head data on the CDK4/6 inhibitors yet, and I hope there never is, because I hope that money is put into studying new agents in new settings rather than to comparing the different agents. And I just think that that would be such a huge, such a huge study. It's hard to know how
the cost of doing the study would be prohibitive, and I agree with you that the findings would be. May not change providers' opinions about which is therapy is best. Now, there can be real-world data that's examined that has some challenges in the coding accuracy, but that might be the best place to identify toxicities and overall survival.
Yeah, I think that it may be for toxicities. I think it's really hard to look at clinical endpoints and real world data in terms of overall survival, although it's certainly been a great interest. I think on the clinician side, we've seen a lot of issues with that data.